Hello everyone, welcome back to our nervous system chapter. So today we'll actually carry forward our discussion about nervous system. In the previous editions of my nervous system videos, I have explained the introduction about neurons, their structures, then types of synapses, and of course types of neurons on the basis of their structures and functions. Okay, so today we are going to actually cover another very important topic from this chapter, that is nerve impulse generation and conduction, where we will understand what exactly is the meaning of the word nerve impulse, how is it generated, and how is it conducted. Okay, so for the nerve impulse generation and conduction, let's basically begin with our quick revision of structure of the neuron, because to understand impulse generation and conduction we'll be requiring knowledge about what? The structure of the neuron, of course. Okay, so as you might recall, the neuron structure consists of what? The sensory areas called the dendrites, which will join to form the dendrons, and then the dendrons would actually give rise to, or sorry, connect to the cyton, and then the cyton would actually continue forward in the form of the single long process that is called the axon, which will ultimately end in the form of axon terminal. So all this actually is done in the previous lecture, structure of the neuron, and if you want to actually get further clarity on it, please refer to the previous lecture about the neuron structure. This is the place where we have the chemicals called the neurotransmitters, and the neurotransmitters are always covered by lipid membrane. The lipid membrane covered neurotransmitters are called the synaptic vesicles, right? And of course we have those gray colored particles here in the cyton as well as in the dendron. These gray colored particles are our Nissel's granules. Okay, so in this neuron we can see that the sensory part or the efferent part of the neuron, sensory or the efferent processes of the neuron, who are those? Dendrons. While the motor, which means the process that is going to take the information or the impulses forward, the motor or the efferent process, there is only one, and who's that? The axon. Okay, so now what exactly is a nerve impulse, how is it generated, and how is it conducted? To understand that, first of all we need to actually understand what exactly is the nature of a neuron when it is not performing any activity, when it is at rest. Okay, so the cell membrane of a resting axon. Resting basically means an axon that is not doing anything right now. I mean a neuron that is at rest. Okay, non-functional for now. The cell membrane of a resting neuron has, or you can say is, charged. Okay, when I say charged, what does that actually mean? It has a potential called resting potential on it. It is a resting potential, and what's the value of the resting potential? Very less, a very mild potential, minus 70 millivolt. So when I'm saying it has got a resting potential of minus 70 millivolt, what I'm referring to is the inside is negative, the outside is basically what? Positive. So this is basically your resting potential. What is the value of the resting potential on both sides of the membrane? The value of the resting potential is minus 70 millivolt. So let's first understand, why will the resting membrane be charged? Why will it have a resting potential on it? There are various reasons behind existence of the resting potential. Let's understand them. The reasons for existence of the resting potential. So what are these reasons? First of all, the presence of an ATPase pump. ATPase pump basically is a protein which is functional, or enzyme we can say, which is functional on all biological membranes of all living cells at all times. Okay, it's a protein or enzyme functional on all biological membranes at all times. And what does it do? It consumes one ATP molecule, and with the help of this one ATP molecule, or the energy obtained from the ATP molecule, what does the ATPase pump do? It transports out 3 sodium ions and transports in 2 potassium ions. ATPase pump consumes 1 ATP to transport out how many sodium ions? 3, and transport in how many potassium ions? 2, creating excess sodium buildup in the ECF, extracellular fluid. If you have read about it anywhere in your studies till now, even in biomolecules chapter, this concept is there, that outside the cells in the extracellular fluid, sodium ions are always in high quantity, while intracellular, who is always in high quantity? Potassium. So now you see the reason behind that, creating excessive sodium buildup in the ECF and excessive potassium buildup inside the cell. Inside the cell where? In the cytoplasm. So if you want to basically talk about, let's say, the representation of an axon membrane here, maybe there is always the operation of a protein pump that is called the ATPase pump. ATPase pump is consuming 1 molecule of ATP, and per ATP molecule consumed, the ATPase enzyme is going to basically transport out how many sodium ions? 3. And at the same time, it is going to basically transport in how many potassium ions? 2 per ATP molecule consumed, which creates excess potassium level buildup inside and excess sodium buildup outside. Who's responsible for this? The ATPase pump. So we'll just understand step by step, one by one, and ultimately we'll reach to the conclusion why there would be a potential of -70 millivolt on the resting membrane. So ATPase pump, as you can notice, is consuming an ATP molecule, transporting out how many sodiums? 3, transporting in how many potassiums? 2. Alright, so that is one thing. Second thing, all biological membranes, including this exolemma, the biological membranes, including the exolemma that we have drawn over here, are more permeable to potassium ions than sodium ions. They are more permeable to potassium ions and less permeable to sodium ions. How does this affect us? See, whenever there is excessive potassium buildup inside, do you think potassium will try to come out by diffusion? Yes, because anything that is present in high quantity anywhere will try to move along the concentration gradient to the place where it is present in less quantity. So potassium ions diffuse out, but what would be the speed of diffusion of the potassium ions? Rapid, because the biological membranes are more permeable to potassium ions. So potassium will start diffusing out rapidly. At the same time, do you think sodium will also try to go in? Yes, sodium will also try to diffuse in. So sodium ions diffuse in, but what would be the speed of diffusion of sodium ions? Comparatively slow. So potassium ions diffuse out rapidly while sodium ions diffuse in slowly. Why? Because the membranes are more permeable to potassium than to sodium. So now understand the combined result. How many positive ions are going out? 3. In return, how many positive ions are coming in? Only 2. Whatever positive ions come in will go out rapidly. Whatever positive ions are going out will stay out only, because अंदर जाने का स्पीड क्या है? स्लो. सोडियम बाहर आ रहा है, बाहर ही रहेगा क्योंकि अंदर जाने का स्पीड स्लो है. पोटेशियम अंदर जा रहा है, अंदर रुकेगा नहीं, रैपिडली बाहर आ जाएगा क्योंकि डिफ्यूजन का स्पीड क्या है? फास्ट है. तो ओवरऑल यहाँ से भी क्या समझ में आया? पॉजिटिव आयन के अंदर जाने का स्पीड क्या है? कम. बाहर जाने का स्पीड क्या है? फास्ट. अंदर 2 पॉजिटिव आ रहे हैं, बाहर 3 पॉजिटिव आ रहे हैं. तो कैन यू सी ना, outside will start turning comparatively more positive compared to what? Inside. Okay, so biological membranes are more permeable to potassium ions than to sodium, hence potassium ions diffuse out rapidly, and at the same time sodium ions diffuse in slowly. This creates a positive ion excess outside. But that's not all. There are other reasons also behind existence of the resting potential. What are those reasons? In the ECF, that is the extracellular fluid around the axon here, calcium ions are also present. Where do you think the calcium comes from? Who is the source of calcium? Blood. So let's say this is your axon. There's a blood vessel maybe over here going nearby. What escapes out of the blood vessel is calcium ions. And where will those calcium ions come? Here. Calcium ions are there in the ECF. Who is the source of calcium ions in the ECF? Blood. And one last thing, D. What is present inside the cell here? Various types of proteins. All these proteins which are present inside your exoplasm or the cytoplasm of the axon or the cytoplasm of the neuron, we can say, they are all rich in negatively charged anionic amino acids. So the proteins of neurons are rich in negatively charged or anionic amino acids. So what would be the overall charge on the proteins? Negative. So look at all the pictures, the entire picture now. How many positive ions went out? 3. Whatever positive ion comes out stays out because अंदर जाने का speed क्या है? बहुत ही slow है. But जो positive ions अंदर गए थे, they are also going to come out only. So already membrane के ऊपर बाहर हमारे पास positive charge और अंदर negative charge in comparison develop होना शुरू हो गया था. Plus ऊपर से बाहर क्या है? Calcium भी है और अंदर जो proteins हैं वो basically क्या है? Negatively charged. So we see where the story is going. What is the combined result of all these things? The combined result of all these things is this, that the inside of the membrane will be always negative and the outside will be comparatively what? Positive. So that's your resting potential, that's your resting membrane that is charged, and the potential on the resting membrane is called resting potential. How much is the value of resting potential? Minus 70. Why is the resting potential existing? We can see that in front of us with these 4 reasons. Number 1, ATP is pumped. using an ATP, throwing out 3 sodium, bringing in 2 potassium. Whatever potassium comes in will try to go out rapidly. Whatever sodium goes out also tries to go in but cannot, because the membranes are always more permeable to potassium. So 3 positive going out, whatever goes out stays out, because inside movement speed is slow. 2 potassiums come in but they don't stay in, they also move out rapidly. Plus there is calcium and there are proteins. The combined result of all these factors is this here: outside positive, inside negative. So I think you got it, all the factors, right? So proteins are negatively charged, or they are anionic amino acid rich proteins. So the combined result of all these 4 factors, the combined result of these 4 factors is the existence of resting potential. Resting potential exists because of the combined result of these 4 factors. And as long as a neuron is not working, is not functioning, this will be always the charge on the exolemma. So this tells us that the cell membrane of a resting neuron has got resting potential. Why? ATPase pump, difference in the permeability of membrane. This is also called differential permeability of the membrane, that is higher for potassium, lower for sodium. And in the ECF there are calcium ions also. In the cell there are proteins which are negatively charged amino acid rich. So ultimately outside will be positive, inside will be negative. Resting potential will be there. How much? Minus 70 millivolt. Now comes the next important point: collection of an external stimulus. For example, when you touch an object, the touch of the object, temperature of the object. If you are looking at something, the light rays are entering your eyes, they are also a stimulus. Right now you are hearing my sounds, so where are the sound waves going? In your internal ear. They are also acting as what? The stimulus. The food that we eat or the air that we breathe, they also have got certain chemicals in it. Taste buds of the tongue and nasal cavity having the neurons in it, those neurons are going to respond to what? Chemicals. So chemicals also act as what? External stimulus. So collection of an external stimulus at which part of the neuron? Dendrites. Collection of an external stimulus at the dendrites triggers opening of sodium ion channels. What exactly are sodium ion channels? Proteins which allow rapid entry of sodium ions in the exoplasm across the exolemma. Once an external stimulus is collected at the dendrites, it triggers opening of sodium ion channels. Sodium ion channels are nothing but proteins which allow rapid entry of sodium ions in the exoplasm across the exolemma. So who's responsible for opening of the sodium ion channels? The collection of an external stimulus at the dendrites. Now the point over here is that the rapid influx or entry of sodium ions, what do you think will it do to the membrane polarity? Reverse it. Rapid influx of sodium ions reverses the membrane polarity, because what is the membrane inside earlier? ऊपर हमने देखा नेगेटिव. अभी सोडियम आयन इतना रैपिडली अंदर आएंगे कि मेम्ब्रेन पोलैरिटी क्या हो जाएगा? रिवर्स. and the potential of exolemma, which was minus 70 millivolt till now, rises by 100 millivolt. How much is the total rise? 100 millivolt. This 100 millivolt is also called spike potential, because there is a sudden rise, no? From minus 70 to plus 30. I mean, this total rise in potential from minus 70 will be actually raising it by 100 millivolt. तो 100 millivolt से raise करेंगे तो पहले कहाँ पहुँचेगा? 0 पे, and then 0 से आगे कहाँ चला जाएगा? Plus 30 millivolt पे. So this rapid influx of sodium ions reverses the membrane polarity, and the potential of exolemma rises by 100 millivolt spike potential. So if the potential was initially minus 70 millivolt और 100 millivolt का rise हुआ है, तो potential finally settles at what value? Plus 30 millivolt. From minus 70, it will come and settle at what? Plus 30 millivolt, which is called the action potential. What is the original potential called? Resting potential. Abhi jo hamara create hua hai because of influx of sodium ion, that is the action potential. So if one of the most common questions in nervous system, what is responsible for creation of the action potential? That's the rapid influx of sodium ions. How much does the potential change? 100 millivolt. There's a spike of 100 millivolt, and that basically raises the potential from minus 70 to plus 30. Okay, so we can actually show it again in the diagrammatic form like this. Every time I'm drawing the diagram, I'll imagine that you already know the previous situation. So the previous situation that we just discussed was there are 4 factors which are responsible for this kind of a situation. What are those 4 factors? ATPase pump, difference in the permeability of membrane for sodium and potassium, higher for potassium, lower for sodium, allowing a rapid diffusion of potassium but not allowing much diffusion of sodium, then calcium ions in the ECF, and then of course the negatively charged protein inside. They are responsible for the resting potential. So as soon as a stimulus is received, let's say the stimulus is received on the dendrons, because they are the sensory parts. These are the dendrons. There's a cell body, and what is arising from the cell body is the axon. So as soon as the stimulus is collected at the dendrons, structural changes from here till here will result in opening of sodium ion channels. There are proteins over here that allow entry of sodium ions. So these proteins normally remain closed, but as soon as the stimulus is applied, we can notice that the sodium ion channels open up, and because the sodium ion channels open up, this is the depiction of opening of sodium ion channels. Because the sodium ion channels are opening up, sodium ion is going to enter in extremely high quantity, influx of sodium, raising the potential inside. What is it earlier? Negative. But abhi sodium itna zyada number mein aayega, bahar kya hai? Positive. Sodium entry will be so much that the inside will become positive and the outside will become negative. Okay, and this potential now is how much? Plus 30 millivolt. So we saw how much rise occurs from minus 70 till plus 30, 100 millivolt ka spike. And who's responsible for generation of the action potential? Rapid influx of sodium ions. Now what exactly was your resting membrane or the resting exolemma called? The resting exolemma has got a resting potential on it, and what is it? Charged, When I say charged, the other term that I can use for charged is polarized. The resting membrane or the cell membrane of a resting neuron is charged or polarized with a resting potential of how much? -70 मिलीवोल्ट. अभी क्या हुआ? स्टिमुलस कलेक्ट हुआ. स्टिमुलस कलेक्ट होने से सोडियम आयन चैनल्स खुले और सोडियम आयन इतना ज़्यादा नंबर में चला गया कि अंदर का पोटेंशियल -70 से राइज होकर +30 चला गया. ओरिजिनल पोटेंशियल -70 था रेस्टिंग पोटेंशियल, अभी नया पोटेंशियल है +30 एक्शन पोटेंशियल. एंड द एक्सोलेमा, द सेल मेम्ब्रेन ऑफ द एक्सॉन, जहाँ पे ये सब चल रहा है, it is now said to be depolarized. What was it earlier? Polarized with a potential of how much? -70 millivolt. बट अभी क्या हो रहा है? बेसिकली सोडियम आयन इतना ज़्यादा अमाउंट में एंटर करेंगे कि मेम्ब्रेन का जो पोटेंशियल था वो रिवर्स हो गया, माइनस से प्लस में चला गया. टोटल राइज हो गया 100 मिलीवोल्ट का, दैट इज़ कॉल्ड स्पाइक. एंड फाइनल पोटेंशियल सेटल हो गया हमारा कहाँ पे? +30 millivolt action potential पे. The membrane is now called depolarized. Now one more interesting fact over here. As long as the stimulus is collected, sodium will keep entering in. You're getting my point? As long as the stimulus is being received on the dendrons, the sodium ions will keep entering in, and as the sodium ions keep entering in, the potential will rise from -70 to +30. But have you ever noticed why does the resting potential never rise above +30? सोडियम तो अंदर आता ही रहेगा ना जब तक स्टिमुलस अवेलेबल है. या तो स्टिमुलस हटा लेंगे तब सोडियम आना बंद हो जाएगा, अंदर आना बंद हो जाएगा. बट एज लॉन्ग एज स्टिमुलस इज अवेलेबल, सोडियम विल कीप कमिंग इन. सो व्हाई इज द पोटेंशियल नॉट गोइंग अबव +30 मिलीवोल्ट? एनी आइडिया? आई विल टेल यू. That's what I am here for. The action potential never rises above +30 millivolt because at +30 millivolt action potential value, at +30 millivolt action potential value, potassium ion channels open. And where is potassium in high quantity? Inside. Where was potassium in high quantity? Inside. Even though they were diffusing out rapidly also, but then the pump is bringing them back again. So there is always an excess of potassium where? Inside. So at +30 millivolt action potential value, the K positive channels also open. So I can actually show the K positive channels like this maybe. These also now open. So here I am showing the opening of potassium ion channels. तो पहले कौन खुला है? सोडियम आयन चैनल इन रिस्पॉन्स टू कलेक्शन ऑफ स्टिमुलस. उससे होगा एंट्री ऑफ सोडियम, और एंट्री ऑफ सोडियम से पोटेंशियल हमारा चेंज हो गया. -70 से कहाँ चला गया? +30. Then once sodium has entered in such an extent that the potential rises to +30 millivolt, at that time potassium ion channels will also open, and exit of potassium occurs through them extremely rapidly. ओके. +30 मिलीवोल्ट पे आके पोटेशियम चैनल्स भी हमारे खुल गए, और पोटेशियम चैनल के ओपनिंग से क्या हुआ? पोटेशियम बाहर जाना शुरू हो गया. तो अब इसका मतलब क्या हुआ? इस पॉइंट के बाद जितना सोडियम अंदर आएगा उसको बैलेंस कौन कर देगा? पोटेशियम का एग्जिट. जितना सोडियम अंदर आएगा, जो कब तक अंदर आता रहेगा? जब तक स्टिमुलस एप्लीकेबल है. बट एंट्री ऑफ सोडियम को नेगेट कर रहा है कौन? एग्जिट ऑफ पोटेशियम. किस टाइम के बाद? इसके अचीव होने के बाद. तो क्या इसके ऊपर पोटेंशियल जाएगा फिर? नो, दैट्स इट. एट +30 मिलीवोल्ट दैट इज एक्शन पोटेंशियल. पोटेशियम आयन चैनल खुल गए हैं. एग्जिट ऑफ पोटेशियम थ्रू देम अकर्स एक्सट्रीमली रैपिडली, बैलेंसिंग और नेगेटिंग any further entry of सोडियम, नॉट स्टॉपिंग, बैलेंसिंग, सोडियम अंदर आता रहेगा बट वो बैलेंस हो जाएगा पोटाशियम के एग्जिट से. सो the action potential remains fixed. At what value? +30 मिलीवोल्ट, क्योंकि एक बार +30 मिलीवोल्ट अचीव हो गया तो सोडियम जितना आगे उसके बाद आएगा उसको पोटाशियम के एग्जिट से हम क्या करते रहेंगे, बैलेंस आउट. सो दैट इज़ वाई रेस्टिंग पोटेंशियल -70 था बट एक्शन पोटेंशियल रेज हुआ सोडियम के एंट्री से. सोडियम एंट्री की वजह से एक्शन पोटेंशियल जनरेट हो गया, +30 मिलीवोल्ट हो गया -70 से. बट विल इट गो बियॉन्ड दैट ऑल्सो? नो, sorry for that. The action potential remains fixed at what value? +30 millivolt, because of the exit of potassium ion through the potassium ion channels which open. So at +30 millivolt action potential value, potassium ion channels open, so the action potential remains fixed at +30 millivolt only. ठीक है, कोई बात नहीं, जनरेट हो गया तो क्या, तो ये कि ये जो एक्शन पोटेंशियल है यही क्या है, impulse. This action potential itself is what? The impulse. So when we say nerve impulse generation and conduction, this is generation. The action potential is called impulse and its generation is called impulse generation. Generation of the action potential is called impulse generation. So that is the first part here of our lecture today, nerve impulse generation. This is your resting membrane. As soon as the stimulus is collected, sodium ions enter in. Entry of sodium reverses the polarity, generates what, action potential. But why doesn't action potential go above +30 even though sodium keeps entering in? Because at +30 potassium exit begins and that never allows the potential to go above +30 millivolts. So action potential will be always how much? +30 millivolt. And this action potential itself is what? Impulse. So creation of action potential through entry of sodium ions, that is impulse generation. Now moving on to the next part, conduction. So what exactly is the conduction of this impulse now? Let's see that. The conduction of impulse. This is extremely simple if you know a bit of physics, which I don't know much, but I'm sure you know it more than me. Here, these are the dendrons. Here's your axon on which I have created the action potential of +30 millivolt. Okay, this is, let's say, the part of axon on which action potential or impulse has been generated. Part of axon on which action potential or impulse is generated because of entry of sodium ions. Now I have to basically take this action potential forward till what? These places, because इधर कौन है, न्यूरोट्रांसमीटर जिसको मैंने रिलीज करवाना है. So I have to make sure that this potential travels forward. Now traveling of potential is basically what? An extremely easy process. Let me tell you how. This much part has become what? डीपोलराइज्ड, इसके ऊपर क्या जनरेट हो गया है, एक्शन पोटेंशियल। बट इसके आगे का एरिया अभी क्या है, डू यू थिंक आगे अभी भी अंदर नेगेटिव चार्ज है और बाहर पॉजिटिव चार्ज है? यस, क्योंकि पूरी मेम्ब्रेन पे एक साथ सोडियम आयन चैनल नहीं खुलेंगे ना। जब भी कोई स्टिमुलस कलेक्ट हुआ ऑन द डेंड्रॉन, तो यहाँ पे बेसिकली आपके जो सोडियम आयन चैनल्स थे वो खुल गए एक्सॉन के स्टार्टिंग पॉइंट पे। मान लो सोडियम आयन की एंट्री हो गई, उसकी वजह से अंदर पोटेंशियल हो गया पॉजिटिव, बाहर क्या हो गया, नेगेटिव। तो ये तो सिर्फ उसी लोकेशन पे हुआ ना जहाँ पे सोडियम चैनल खुले हैं, पूरी मेम्ब्रेन पे थोड़ी एक साथ सोडियम चैनल खुल जाएंगे। स्टिमुलस कलेक्शन के बाद सबसे पहले सोडियम आयन चैनल आपके एक्सॉन के स्टार्टिंग पॉइंट पे ही खुलते हैं, तो वहीं से क्या शुरू होता है हमारा, डीपोलराइजेशन। एक्सॉन के स्टार्टिंग पॉइंट का नाम क्या है वैसे बाय द वे? द स्टार्टिंग पॉइंट ऑफ द एक्सॉन इज कॉल्ड एक्सॉन hillock. Axon hillock is the starting point of the axon. Also considered as the place of generation of impulse or the place of generation of action potential. तो एक्शन पोटेंशियल हो गया जनरेट, या इम्पल्स हो गई जनरेट। ऑन विच पार्ट ऑफ द एक्सॉन? द एक्सॉन हिलॉक पार्ट, द फर्स्ट पार्ट वेयर सोडियम आयन चैनल्स ओपन फॉर द फर्स्ट टाइम। So now back to this picture. This is the portion where entry of sodium has created the action potential, and we can notice that on the rest of the membrane अभी भी क्या है, there is still a resting potential only. Resting potential still where? On the rest of the exolemma or the membrane of the एक्सॉन, उसके ऊपर क्या है, अभी भी रेस्टिंग पोटेंशियल ही है। जबकि इनिशियल पॉइंट पे हिलॉक पे क्या जनरेट हो चुका है, एक्शन पोटेंशियल। इधर अभी भी क्या है, रेस्टिंग पोटेंशियल। तो डू वी एक्चुअली नीड टू डू एनीथिंग फॉर द करंट टू ट्रैवल फॉरवर्ड और द एक्शन पोटेंशियल टू ट्रैवल फॉरवर्ड? नो। इफ यू नो फिजिक्स, एज आई वाज सेइंग, current travels from positive towards negative charge, and the circuit is completed by the same current traveling outside from positive to negative charge. When this happens, look at this carefully. The current travels from positive charge to negative charge, converting this negative charge into what? Positive. And इधर पीछे बैक साइड पे क्या हो जाएगा, नेगेटिव। यू गेटिंग माई पॉइंट? करंट ट्रेवल्स फ्रॉम हियर टिल हियर इन दिस डायरेक्शन। सो व्हेन करंट इज ट्रैवलिंग इन दिस डायरेक्शन, तो जो आगे का नेगेटिव चार्ज था वो पॉजिटिव में कन्वर्ट हो गया, और जो पीछे का पॉजिटिव था वो नेगेटिव में कन्वर्ट हो गया। ऊपर भी देख लेते हैं, यही हो रहा है। ऊपर ये हो रहा है कि भाई जो करंट के ट्रैवल की डायरेक्शन है उसकी वजह से पीछे बैक साइड पे क्या जनरेट हो जाएगा, पॉजिटिव चार्ज, और आगे यहाँ पे क्योंकि पॉजिटिव चार्ज पीछे की तरफ मूव कर रहा है, तो इधर क्या क्रिएट हो जाएगा हमारा, नेगेटिव चार्ज। करंट ट्रेवल्स फ्रॉम पॉजिटिव टू नेगेटिव, पॉजिटिव टू नेगेटिव। बिकॉज ऑफ द ट्रैवलिंग ऑफ दिस करंट, पॉजिटिव चार्ज आगे शिफ्ट हो गया इधर, ये पॉजिटिव चार्ज पीछे शिफ्ट हो गया इधर, और बाहर क्या हो गया, नेगेटिव। नाउ लुक एट दिस केयरफुली। अर्लियर व्हाट वाज द कंडीशन हियर, बाहर पॉजिटिव अंदर नेगेटिव, पर क्या वो रिवर्स हो गया है, बाहर नेगेटिव और अंदर पॉजिटिव? यस। तो ये जो हमारी कंडीशन थी कौन सी वाली, एक्शन पोटेंशियल वाली। जब मैंने अंदर का चार्ज पॉजिटिव में कन्वर्ट कर दिया यहाँ से मूव करा के, बाहर का चार्ज नेगेटिव में कन्वर्ट करा दिया पीछे मूव करा के, हो गया ना, ये रहा डीपोलराइजेशन। भाई डीपोलराइजेशन था क्या, बाहर नेगेटिव अंदर पॉजिटिव, तो ये हो गया बाहर नेगेटिव अंदर पॉजिटिव। तो एक्शन पोटेंशियल जनरेट कराना है आपने कहाँ पे, हिलॉक पे, सोडियम आयन चैनल्स खोल के। एक बार एक्शन पोटेंशियल जनरेट हो गया तो उसके बाद ट्रैवलिंग इज सिंपल फिजिक्स का लॉ दैट करंट ऑलवेज ट्रेवल्स फ्रॉम पॉजिटिव टू नेगेटिव चार्ज। यहाँ पे एक्शन पोटेंशियल जनरेट हो गया, अभी आगे जनरेट हुआ नहीं है, तो ये खुद ही यहाँ ट्रांसफ़र हो जाएगा। सिमिलरली जब यहाँ पे पॉजिटिव चार्ज और पीछे नेगेटिव है तो ये पीछे ट्रैवल करेगा इधर, इससे इसको हम कहते हैं कि भाई सर्किट कंप्लीट हो गया करंट फ्लो का। बट अल्टीमेटली हुआ क्या, दो चीज़ें हुई इसमें, देखो इंटरेस्टिंग, आई टेल यू व्हाट। सबसे पहली चीज़ तो ये कि जब ये वाला पॉइंट आपका डीपोलराइज था तो ये पॉजिटिव चार्ज के आगे और ये पॉजिटिव चार्ज के पीछे मूव करने से यहाँ हो गया पॉजिटिव, यहाँ पे क्या हो गया नेगेटिव, तो इधर अपने आप क्या ट्रैवल जनरेट हो गया, एक्शन पोटेंशियल या करंट या जो इम्पल्स आपने यहाँ पे जनरेट करवाई थी वो आगे बेसिकली क्या हो गई, ट्रैवल। प्लस इसका एक और एडवांटेज दिख रहा है आपको, क्या, जब ये करंट आगे ट्रैवल करेगा तो आगे का पार्ट अंदर से क्या हो गया, पॉजिटिव चार्ज, यहाँ पे क्या क्रिएट हो गया आपका एक्शन पोटेंशियल। बट एट द सेम टाइम जब इधर से पॉजिटिव चार्ज पीछे ट्रैवल कर रहा है तो यहाँ पे क्या हो गया पॉजिटिव, और यहाँ अंदर क्या हो गया था, पॉजिटिव चार्ज के आगे जाने से नेगेटिव। तो इधर एक्शन पोटेंशियल जनरेट हो गया एंड साइमल्टेनियसली पीछे मेम्ब्रेन वापस कहाँ पे चली गई, बाहर पॉजिटिव अंदर नेगेटिव, वापस कहाँ पे चली गई, बाहर पॉजिटिव अंदर नेगेटिव, इधर। तो इससे बेसिकली हमारे दोनों चीज़ें सॉल्व हो जाते हैं। आपने यहाँ पे एक्शन पोटेंशियल या इम्पल्स जनरेट करवा लिया, जनरेट करने के बाद कंडक्शन क्या है, आपको सिर्फ पॉजिटिव चार्ज से नेगेटिव चार्ज की तरफ करंट फ्लो कराना है। तो वो अंदर किधर होगा, आगे की तरफ, और बाहर किधर होगा, पीछे की तरफ, अंदर आगे की तरफ, बाहर पीछे की तरफ। ओके, so this is the part of the axon on which impulse is generated, the action potential, and this is the part of the neuron जहाँ पे अभी भी क्या है, resting potential. Resting potential on the rest of the axillemma still exists. On the rest of the axillemma अभी भी क्या exist कर रहा है, resting potential. So current travels inside the axillemma. Forward. Here, forward, and travels outside the axillemma backwards. Current travels forward, backward. Hence, the membrane keeps getting depolarized gradually. The membrane keeps getting depolarized gradually. अंदर नेगेटिव था, यहाँ से करंट ट्रांसफर की वजह से अंदर क्या हो गया, पॉजिटिव। तो मेम्ब्रेन ग्रेजुअली क्या हो गया, आगे डीपोलराइज, एंड एट द सेम टाइम पीछे क्या हो गया, वापस चला गया कहाँ पे, नॉर्मल रेस्टिंग पोटेंशियल पे, जिसको क्या कहते हैं, रीपोलराइज्ड। सो द मेम्ब्रेन कीप्स गेटिंग डीपोलराइज्ड ग्रेजुअली इन फॉरवर्ड डायरेक्शन, मतलब किधर, टूवर्ड्स द axon terminals, and also interesting stuff, keeps getting repolarized. ये हो रहा है यहाँ पे, ये पीछे चला गया पॉजिटिव चार्ज, यहाँ से पीछे, तो इधर क्या क्रिएट हो गया, पॉजिटिव, यहाँ पे क्या क्रिएट हो गया, नेगेटिव। तो साइमल्टेनियसली एक साथ दोनों चीज़ें हो गई, यहाँ पे एक्शन पोटेंशियल जनरेट हो गया और यहाँ पे पोटेंशियल वापस कहाँ चला गया, नॉर्मल जो ओरिजिनल था -70 मिलीवोल्ट। तो बस यही आगे तक करते जाने हैं। ये सारे जो नेगेटिव चार्ज हैं किस में कन्वर्ट हो जाएंगे, पॉजिटिव में। यहाँ पे आगे फिर से क्या है, ये गया करंट इधर, ये गया करंट इधर, तो ये आपका एक्शन पोटेंशियल आगे ट्रांसफ़र हो गया और पीछे का पार्ट साथ साथ में क्या हो गया, रीपोलराइज भी हो गया, उस पर नॉर्मल पोटेंशियल जो -70 था वो रिस्टोर भी हो गया। So the membrane keeps getting depolarized gradually in the forward direction towards the axon terminal and keeps getting repolarized also. That means the potential of the axillemma keeps getting reversed. reverse to what? Minus 70 millivolt. It keeps getting repolarized also. That means the potential keeps getting reversed to minus 70 millivolt in the backward direction. In the backward direction means towards साइड ऑन. बिकॉज सी न्यूरॉन ने कोई एक सिंगल इम्पल्स थोड़ी मतलब सिंगल स्टिमुलस का थोड़ी रिले करना है इन्फॉर्मेशन. थोड़ी देर के बाद अगला स्टिमुलस आ जाएगा, उसको भी रिले करना है, अगला स्टिमुलस आ जाएगा, उसको भी रिले करना है. तो मतलब ये कि एक स्टिमुलस कलेक्ट करके जो एक्शन पोटेंशियल जनरेट किया उसको तो आगे ट्रांसमिट कर दिया, साथ साथ पीछे क्या होता जा रहा है, रीपोलराइजेशन भी होती जा रही है. क्योंकि अगला स्टिमुलस जब आएगा तो मेम्ब्रेन को क्या होना चाहिए, अंदर नेगेटिव बाहर पॉजिटिव रीपोलराइज्ड होना चाहिए. मतलब इसका पोटेंशियल वापस कहाँ जा चुका होना चाहिए, माइनस 70 मिली वोल्ट पे. तभी ये अगले स्टिमुलस को अपने अंदर से ट्रैवल करवाएगा. तो वो भी साथ साथ कर रहे हैं हम लोग यहाँ पे, एज वी कैन सी. ओके, सो वी कैन सी दैट द मेम्ब्रेन कीप्स गेटिंग डीपोलराइज्ड ग्रेजुअली इन द फॉरवर्ड डायरेक्शन. The membrane keeps getting depolarized gradually in the forward direction and keeps getting repolarized also in the backward direction at the same time. ठीक है, तो चलो ये करते करते ये पॉजिटिव चार्ज को आगे ट्रांसफ़र करते करते ये जो हमने किया है यहाँ पे, ये रहा पॉजिटिव चार्ज को आगे ट्रांसफ़र करते करते आप बेसिकली कहाँ पहुँच गए, धीरे धीरे एक्सॉन टर्मिनल्स पे आ गए फाइनली. तो जब आपका एक्शन पोटेंशियल यहाँ पे जनरेट हुआ था वो था इम्पल्स जनरेशन, और ये जो अभी चल रहा है बेसिकली पॉजिटिव चार्ज आगे और इधर पॉजिटिव चार्ज चल रहा है पीछे, ये क्या है, इम्पल्स का ट्रांसमिशन. So the membrane keeps getting depolarized gradually in the forward direction and getting repolarized also in the reverse direction. The process of gradual generation of action potential. The process of gradual generation of action potential on every single part of exolemma from axon hillock towards axon terminals. The process of gradual generation of action potential in every single part of exolemma from axon hillock towards axon terminals. This is called impulse transmission. So what is impulse generation? Generation of action potential, and the process of gradual generation of action potential on every single part of exolemma. One by one, the shifting of the positive charge from axon hillock towards axon terminal, this is called impulse transmission. And finally, the action potential reaches on the presynaptic membranes. Finally, the action potential reaches on the presynaptic membranes. पहुंच गया, ये रहा एक्शन पोटेंशियल प्रीसिनेप्टिक मेम्ब्रेन्स पे आ गया इधर से ट्रेवल करते हुए. The action potential finally reaches on the presynaptic membranes. Positive inside, negative outside. That's impulse generation and impulse conduction. But then this is not the end of the story. What is the impact of generation of action potential over here on the axon terminals? Let's see. The depolarized or on the depolarized presynaptic membranes. On the depolarized presynaptic membranes, calcium ion channels will open. On the depolarized axon terminal membranes, or the presynaptic membranes which have got depolarized just now, what is going to open over here? Calcium channels. So what enters in through the calcium channels? Obviously calcium ions. So calcium ions enter in the synaptic knobs or the synaptic end bulbs. You remember what is this? Synaptic knob or the synaptic end bulb. And what has started coming into it? Calcium ions. Once the calcium ions enter, what will they do? The calcium ions attach. कौन present है अंदर synaptic knobs के? Synaptic vesicles. The calcium ions attach to synaptic vesicles. ये रहे ना यहाँ पे. What are these synaptic vesicles? The membrane bound vesicles. Here. Calcium ions attach to these synaptic vesicles and cause their precipitation on the presynaptic membrane. Let me zoom in into this for you. Calcium ions enter in and take these vesicles and stick them on the presynaptic membrane. Calcium ions attach to the synaptic vesicles and induce their precipitation or attachment, we can say, on the presynaptic membranes. If we are talking about a chemical synapse, and if you don't know what is a chemical synapse, refer to the nervous system lecture 2 which we discussed earlier. If calcium ions enter in and cause the precipitation of the the synaptic vesicles on the presynaptic membrane, who will start coming out by diffusion now? The neurotransmitter chemical. Which will travel across the synaptic cleft and then attaches to what? Post synaptic membrane. The membrane of what? The target organ. That's it. This is what we wanted to do. The neuron was supposed to control what? The activity of the postsynaptic organ or the postsynaptic membrane of the target organ. तो कर लिया इसने. Calcium ions ने क्या किया? इन्होंने trigger किया release of the precipitation of the synaptic vesicles on the presynaptic membrane. So the neurotransmitter chemical comes out by diffusion in the synaptic cleft and travels across the synaptic cleft, attaches to the postsynaptic organ or the target organ, attaches to the target organs postsynaptic membrane to regulate its activity. Once again, the term over here that I mentioned is regulate. Did we explicitly mention or clearly mention increase or decrease? No, because the neurotransmitter chemicals released by the neurons can be actually of two types: inhibitory, which will attach to the postsynaptic membrane of the target organ and stop the activity of the target organ; excitatory, the neurotransmitters that will go to attach to the postsynaptic membrane and stimulate it. So the neurotransmitter chemicals come out of the synaptic cleft, attach to the target organ's postsynaptic membrane to regulate its activity. When I say regulate, increase or decrease, or initiate और stop. अगर पहले से काम चल रहा है तो न्यूरोट्रांसमीटर्स को स्पीड अप करवा सकता है, स्लो डाउन करवा सकता है. पहले से काम चल रहा है तो न्यूरोट्रांसमीटर्स को बंद कर सकता है. पहले से काम चल रहा है नहीं चल रहा है तो न्यूरोट्रांसमीटर्स को स्टार्ट भी करवा सकता है. तो न्यूरोट्रांसमीटर्स क्या करते हैं? दे रेगुलेट. ऑन द बेसिस ऑफ टाइप ऑफ रेगुलेशन दैट दे डू, neurotransmitters can be classified and the name itself will make it clear to you. The neurotransmitters can be classified as excitatory neurotransmitters and inhibitory neurotransmitters. As names are suggesting, excitatory neurotransmitter excites the postsynaptic organ or the target organ. Inhibitory neurotransmitter inhibits the target organ. The examples of common excitatory neurotransmitters that we have in our body: adrenaline, acetylcholine. Examples of inhibitory neurotransmitters: a common one in the brain, that is dopamine, then GABA. GABA is gamma amino butyric acid. So we can see that neurotransmitters are not only excitatory, they can even be basically what? Inhibitory. Okay, so that's it, guys. This was the end of our total discussion today regarding nerve impulse generation and nerve impulse conduction through various pictorial diagrams, and of course the type of neurotransmitters in the end. So we'll stop the lecture over here today, and our next target in the nervous system will be understanding the structure of our brain, the most important part of the central nervous system. So the introductory part of the chapter with neurons and their way, their types, their mechanism of action, all that is over. Neuroglial cells also are of course there in the nervous tissue. Neuroglial cells are helpers and they will be actually covered separately in the tissues chapter. So that's it for now. I'll be actually sharing another lecture with the structure and arrangement of neurons in the brain. Till then, keep revising, keep studying, keep spreading the word about the channel. Like, subscribe and share with your friends also. I'll see you in the next video. Thank you. Bye-bye.